de las actividades y los resultados que muchas de ellas también se realizan en el seno de la universidad. Comentarles que Enrico, a quien van a tener el placer de escuchar, acá sí necesito machete, el doctor Enrico Francia, es profesor de Agronomía y Ciencia de los Cultivos de la Universidad de Módena y Reggio, Emilia, UNIMORE, en Italia. Coordina una maestría en sostenibilidad integrada de los sistemas agrícolas y también participa en programas de grado y doctorado en ciencias y biotecnología agroalimentaria. Se desempeña además como vicedirector del Centro Bioges CITEIA de UNIMORE, Centro de Investigación para el Mejoramiento y la Valorización de los Recursos Biológicos Agroalimentarios. Representa, como les decía, a la Universidad UNIMORE en ámbitos académicos, institucionales y de articulación público-privada a nivel nacional y regional. Y en ese contexto, y como uno de los objetivos también de nuestra universidad es la internacionalización, la visita de Enrico se produce en el marco de un proyecto que está financiado por Italia y por la Unión Europea, que es el proyecto Green and Pink for Sustainable Education, que articula una red en la cual participan 9 universidades italianas y 41 universidades de Latinoamérica, y entre las 2 universidades de Argentina que participan está la Universidad Torcuato Di Tella y la UNNOBA. Así que para nosotros es un orgullo y un placer contar hoy con la presencia de Enrico, que vino en una visita muy corta, conoció las sedes, estuvo en Junín, estuvo en Pergamino, y hoy también preparó una charla para poder contarles algunas diferencias entre lo que se hace en Europa, en Italia en particular, y en torno a los bioestimulantes. Así que, Enrico, un placer. Comentarles también que si hablan más lento, ¿no?, como hablo yo, que hablo a toda velocidad, Enrico entiende muy bien español, así que al momento de las preguntas las pueden hacer en español. Si hay alguna dificultad para comprender, Fiorella, que es de Relaciones Internacionales, o yo o alguien más puede preguntarle en inglés, así que no duden en hacerlo. De nuevo, agradecerles a los organizadores de la jornada, a nuestros docentes, a los investigadores, a todos quienes se acercaron, invitarlos e invitarlas a que se acerquen a la universidad. Es un placer contar con una jornada con esta altísima recurrencia y que hable de nuevas tecnologías en torno a la sustentabilidad. Así que bueno, espero que disfruten la charla y muchísimas gracias.
Muchas gracias. Es un honor para mí estar aquí. Gracias a EnBio, a Pablo, por la invitación, y gracias a UNNOBA, Virginia, por la presentación. Disculpen si la presentación será en inglés, porque mi español es realmente básico. Posso intendere lo spagnolo se voi parlate lentamente, così come voi potete capire l'italiano se io parlo lentamente. Ma passo all'inglese. I switch to the English for the presentation. Okay, so I divided the presentation in 3 parts. The first part will be related to the mechanisms underlying the winter cereal adaptation to climate change in our region, in particular to a topic that is very important for me and for my research group, that is related to frost tolerance in barley, la cebada. Why barley? Barley is a very important species, is a diploid one that could be used also as a model for wheat production, el trigo. It is a very important crop also for South America and for Argentina. This is a typical picture of our region. We are based in the north of the country, el norte de Italia. As you can see, that is a field trial with several varieties, and it happens more and more frequently that we have winter without snow. And when the temperature goes below 0 degrees, you could have this situation on the leaves of the plants. Ice formation and damages, especially for barley, that is, okay, it is a winter surviving species, but with a different level of winter survival capacity compared to the other, let's say, 3TC species in terms of botanics. The three species within the tribe of the 3TC are barley, la cebada, wheat, il trigo, and rye. I don't remember the translation in Spanish for segale. Thank you. So the three species have different levels of resistance, but the resistance, as we have said, as we have seen today's morning, it depends the resistance to a process that is a physiological process, the acclimation. If you don't acclimate the plants, they are not able to resist, to withstand the stress. But you see, even when you cold acclimate the three species, they have different levels of resistance. And there is also another variability. The presence of winter varieties and spring varieties. They have different levels of resistance. The winter varieties are interesting because they require cold to flower, but they also need the capacity to withstand with the cold stress. So they have a higher level of resistance. A bit of molecular mechanism. The molecular mechanism is based on sensing the stress, the ability of the cell of the plant to understand that cold is coming. You remember the series in Netflix, The Winter is Coming. It is very important to sense the stress and to inform the nucleus. And in the nucleus there will be the effector genes able to produce the resistance. That is the cold acclimation. And there are a lot of cold activated genes, but the very important responsible of this resistance are transcription factors. Even today morning we heard something about i regolatori della trascrizione. And some of them are very important and they are called CBF genes. I will talk about that later. In my research group we started exploring the natural variability in terms of capacity to withstand the cold, and we developed a biparental population. We selected two varieties. One, the blue one, that has the name of a river that is close to my house, Nure. It is a winter type, it is resistant. And the green one has a French name, Tremois. It is a spring variety susceptible to cold. And the two varieties are very interesting. The blue one, let's say, is used for feeding. You know that barley has multiple attitudes. We can eat barley. Animals can be fed with barley, the blue one, but also barley varieties can produce a good malt to produce beer. And we were able to find in this segregating population that there are two genes responsible for frost resistance. And the two genes, interestingly, are very close on the same chromosome. And what are the genes that are present under these two chromosomes? One is responsible for vernalization requirement, so the plant needs to feel cold to flower, and then they are associated with frost resistance. I have to feel cold, but I have to be prepared to resist. And we were lucky, we published a lot of papers, I don't want to bore you about this, but we identified a very long list. Not just one transcription factor, but a series of transcription factors. 13, at least, in the barley genome. So, a first question came into our mind. Is it a matter of copy number? The more transcription factors we have, the more higher the resistance of the varieties. The lower the number of copies, the lower the resistance. And we selected a population of cultivated barley with different origin, different ability to withstand the cold, and we tested them in the field, and we tested them molecularly to find the copy number variation C and V of the CBF genes. And you can see here that the varieties with an higher level of resistance cluster in this part of the graph. The middle standing varieties are in the middle, and the spring one with a lower level of resistance are here with a single copy. And then we were able to study the genetic code, the sequence. I remember you that the barley genome is very large, is as large as the human genome, 3.5 gigabases, very large, but a fewer number of genes. That means, it is too complicated, I know, but just look at the colors. Look at the colors. The genes are the triangles, and the rectangles, blue and red, are just repetitive elements. Something that we call junk DNA. Nothing useful, but it's not true. It is also useful. So we were the first one to sequence the entire portion harboring the CBF genes in chromosome 5 in the reference cultivar Morex that is susceptible, but it is very good for malting production, and it was for many years the reference standard in the US for the malting barley production. And then we did the same with the Nure variety. We constructed a genome library, and we were able to sequence the chromosome. So we have the two sequences in comparison, the blue one and the green one, the resistance and the susceptible varieties. And then we can compare them. And if we look at the sequence in the two varieties, we can find that there are different genes. Some variety has more genes and the other lower number. And then we found the copy number variation. This portion that is in the susceptible Morex has 2.5 copy in Nure. So it seems that the copy number variation is the basis for this natural ability to withstand cold. We are also preparing, if there are geneticists inside, the QTL NILs, quantitative trait loci near isogenic lines. It means lines that are very, very similar, siblings in a sense, and the only difference is that we introgressed, a better term, introgressed a portion of the genome that is the only one that is different. So we can see the effect of having only that part in a common background, because it's the background that makes the difference. And then we moved to the field, not only in the growth chamber, but also to the field. And in this case, again, we studied Nure and Tremois, and we studied a lot of genes. During the development of the plants, and we studied the CBFs, the regulator, and also the effector genes. You remember we have the opposite situation for the winter and for the summer. So for us, December and January are the coldest months. You can see here the hardening threshold means when the plant is sensing the cold that is coming. And the frost threshold is the stress. And we have the sampling week by after week, sampling the leaves of the plants and then extracting the RNA. But we also tested the chlorophyll content of the plants using the SPAD. Many of you are used to test the chlorophyll content, and you can see we have Nure, we have Tremois and other varieties, but the resistant one is always the darker green. All over the period, even if you have some events with the snowflakes, that means that the temperature went under zero, below zero. So this is just an example of expression. I don't want to bore you, because I have many things to say. So what happens if you just look at the blue and the red color? Blue means the resistant and the red means the susceptible. And all the histograms are a time series, starting from December 17 to now, 25 of February. So you see, for this regulator, the blue one wins at the beginning of the period. And again for the CBFs, Nure for CBF2 and for CBF4 has an early capacity to hear that the cold is coming. Of course, plants don't have ears, but they can feel it. And then again, sometimes when it is too late, the red one starts to push up the expression of some gene. And this is the situation of the effector gene. So the effector gene, of course, are late respect to the peak of expression of the transcription factors. So it is obvious that we see Nure winning this race later, after the CBFs were expressed. I can skip this one about vernalization requirement because we have data that say that Vern1 is able to push down the expression in the spring genotypes. This situation was expected. But I want to come to this first conclusion. I put this because many of you, okay, there is an Italian talking on a palco, and of course I have to speak about opera. I don't see Pablo. Pablo told me that his own, the grandfathers of Pablo are coming from a little town that is 30 minutes driving from my house. So, and that specific area of the north of Italy gave the birth to Maestro Verdi. Many of you know the opera of Verdi. And this is Maestro Muti. This is my explanation for what I said. Of course, if you are in a big orchestra, there are at least 52 maestro. Everyone can play more than one instrument. The violin, the piano, the trumpet. They studied years and years, 8, 10, 12 years. But to have an orchestra playing an opera, you just need a very skilled maestro, like Maestro Muti. Only in that way you will have a very nice opera. And that is the meaning of the transcription factors. If you have a very good transcription factor, as the CBF genes are, you can have a good response from the plant. And then I move to the second topic that is more related to today's meeting. That is the role of soil microorganisms. I'm not a microbiologist, but we did something in agronomy isolating bacteria from tomato rhizosphere. You know, tomato is a very important crop for our region. We have tomato in crop rotation with maize and with wheat and barley, because we crop barley in tomato in open field for canning tomato production. We are in competition with the Californian farmers, so it is an hard fight for us. But we isolated some rhizosphere bacteria from the root of tomato, and we have seen a very good response of the tomato crop in response to the inoculation with this bacteria. And our question was, what are the ability of these bacteria that we have isolated? All of them are able to fix the nitrogen from the air. Some of them are producing cytophore in the membrane, and some of them are producing indole acetic acid, that is a very powerful hormone. And the first question for us was, can this microorganism isolated from the tomato rhizosphere also benefit the barley crop? So we started as usual in the lab, and we identified one that is very interesting, a Pseudomonas chloraphis aerophaciens, with a very good performance. Then we moved to the greenhouse, testing again, more or less you see the same name of the varieties, looking if they have some capacity. For example, for drought stress. So we were using these treatments. This was used as the control, 40% of the soil water storage capacity, and we imposed a stress at 20% of the soil water storage capacity. And we measured a lot of parameters, water use efficiency, physiological, morphological, and agronomical traits, leaf chlorophyll content, and so on. But you can see here, I draw your attention to this line. Yes, the inoculation was very effective. So you can use a microbes that is important for a species and that can be effective also for another one. This is very relevant because we have those species in crop rotation in our fields. We can inoculate multiple species with the same bacteria. And then we move to the field, of course. Again, the same genotypes, again, the same treatments. In this case, we did something different about the nitrogen. So we started, all the plots were at 70% of the nitrogen requirement of the barley crop, and this amount was given through cow manure. And this was the control. And then we added the remaining 30% with mineral fertilization, the middle line, and then we added the bacterium without the mineral fertilization. And you see the results. So the yield production, that is an average yield production for us in our region, is boosted by the bacteria. And you can save 30% of the nitrogen input by fertilizers, of course. So the last minute I will skip to something that maybe is interesting also for you to have a look to what happens in the other hemisphere across the ocean. But I know that you have many parents and families there. So el maíz, corn, is also a relevant species also for us in Italy. And as you can see the situation, Italy has a sort of corn belt as they have in the US, because it is the situation with much more water. But we have a lot of problems, and maybe this could be an opportunity for the Argentinian. You see what are our problems. Of course, as all over the world, when they look at the red line, when the hybrids were introduced in maize cultivation, the production was boosted. This is 1.7 quintals quintal per year. I'm sorry if they are in Italian, this one. But something happened recently, and you see this. We reached a plateau of productivity. Okay, and we have the environmental influence. Some years is better, and some years are very bad. Like this one, like this one. Things are going worst, because the red line are the acreage of maize in Italy, and you see they are going down, down. We reached, last year, we reached the minimum for the Italian acreage dedicated to maize. And also the production, the productivity, the total production in terms of million tons is going down. What does it mean? Okay, this one. Of course, we have national trials. You can see what happens and what is different. I don't really know the comparison with the Argentinian situation, but you see, for the long cycle maize hybrids, we harvest at a very high relative humidity, and then we have a step of essiccation, drying, also to reduce the incidence of fumonisin. And then you see the 600 class FAO and so on, and that is the production, and this is the same for the early and intermediate maturity cycles. This is the average yield, el promedio de rendimiento. Across the years, you see there are years that were terrible, like 2003, no rain at all during the whole summertime, and not all the maize surface in Italy are irrigated. Okay, again, this is the situation, but this is our problem. We are net importers of maize because we are going down with the surface and with the production. We are importing mainly from Ukraine, and this was another problem when the war happened in 2022. And you see, mainly we are importing from the east of Europe, eastern part. But what are the trends worldwide and in Europe also? You see, we can see altogether that the China line in terms of consumption is always green. It means that year by year by year they are increasing the demand of maize, of corn. And they are driving the increasing demand in estimates and forecast for the world. But in Europe we are going down, and I'll tell you why. For two reasons. The first reason is that many countries, I would say almost every country in Europe except Spain and to a little extent Portugal, we are lagging in terms of genetics. You see here, this is the environmental effect. This wobbling up and down, year by year. And why in Spain they have a more stable line and very high productivity? Because they started to cultivate GM crops. But we don't. This is crazy. We cannot cultivate GM crops, but we have, absolutely, we have to import them. But this is another chapter. This is another story to explain this to the politician, but forget it. Please don't record me for this part. What happens? The production of maize is moving. is moving east, is moving toward Hungary, Romania, Serbia, because they have lower cost of production. But also it is moving north because of the climate change. So the Polish are very happy that the environment is warmer, because they started to grow maize and to sell it to Italians and French also. Okay, I'm going to conclude because maybe you are very tired. And my conclusion is general. This is the picture of my country and my region. Pablo, you are here. So, just look at the colors. You see the colors. They mean that we have a very variable intensification. We have rural area, the green one, with intensive agriculture and very high specialization. And then we have the blue one, area with some problem of development. It means all the blue parts you see, they are corresponding to the Alps and Apennine mountains. Of course, population is going down, people is moving from the mountains to the plain because they are site of opportunity. We have very good transportation, large cities, internet connection, and the young people is moving. And then the yellow one is the middle way. And you can also see how small we are compared to you. So the total cropping area of my country is 12.5 million hectares. Honestly, I don't know what is the Argentina, 35, 40, okay. One third. But we have the problem of the use of fertilizers and chemicals also. So we must be more sustainable. And we are facing this environmental challenge as everybody, but maybe in the Mediterranean, that is a very close sea, not so deep, we are feeling this change. These are, if you compare the average temperature of the 91 to 2020 compared to the previous 30 years, 61 to 90, this is the situation, more or less 1.5, 1.2 degree more. And this is the anomaly trends. So the last one, another one. And these are the projection for the future. Maybe we will have no change in total precipitation, but some events with a very high raining period and dry continuous period. So this is the last one. So to summarize everything that was told today, we have to put together three major factors. Every agronomist, every geneticist knows it, but we have to repeat it every time. What is resa is the yield, rendimento, is a triple interaction between the environment that gives us a quantity of resources, a Q, and it is the interaction with the genotype. You have to choose the best genotype and put them in the best situation. How? With the agrotechnics, with agronomic management, to reduce the crop stress index. Otherwise, if you have a limiting factor in the green, in the red, and in the blue part of the formula, the yield will be very low. So this is our challenge for the future. Work together, the agrometeorologist, the geneticist, the microbiologist, and the agronomist to maximize this formula. So this is some acknowledgement to my group, and muchas gracias for your attention.